Welcome back to this next video in which we are discussing influenza. Okay, so we're currently in the process of discussing the adaptive immune response to influenza. And remember the spirit with which we're doing this. We want to see the bare skeleton of the adaptive immune response. We want to see the big picture rather than the in-detail discussion of the adaptive immune response. So, the big picture is that we're going to initiate two different types of adaptive immune response to influenza viruses. We're going to initiate a B-cell response, which is a humoral adaptive immune response, and that's a response to the extracellular influenza viruses. And we're going to initiate a cytotoxic T lymphocyte response, which is an example of a cell-mediated response, and that's a response to the intracellular influenza viruses. Now, at the moment we're in the process of discussing the extracellular response, so remember all adaptive immune responses start with antigen-presenting cells, of which the two major examples are dendritic cells and macrophages, particularly dendritic cells. They're fantastic at doing antigen presentation. So we're starting with the extracellular response. What will happen is these antigen-presenting cells will phagocytose extracellular influenza particles. They will thus know it's an extracellular pathogen, and they will take that decision, let's initiate an extracellular adaptive immune response. So what will happen is they will phagocytose the influenza virus particle, they'll break up all of its antigens into little peptide fragments, and the way that they break down its proteins into fragments is random, so different antigen-presenting cells breaking down the same antigens will break them down differently. So overall what you will end up with is loads of antigen-presenting cells that are covered with MHC class II complexes, whoops, get me the laser pointer, MHC class II complexes with peptide fragments of antigens of influenza viruses in those peptide binding grooves of the MHC class II. And remember, MHC class II is what we put peptide fragments of antigens from extracellular pathogens on. Okay, so what will then happen is the antigen-presenting cell, or antigen-presenting cells, because it's going to happen in the bulk, loads of antigen-presenting cells covered in MHC class II peptide fragment complexes are going to go to the lymph nodes where they want to find CD4 positive T cells. So the type of cells that can be activated in the extracellular response are the CD4 positive T cells. There are two populations of T cells fundamentally, CD4 positive T cells and CD8 positive T cells. CD4 positive T cells are part of the extracellular response, CD8 positive T cells will be part of the intracellular response, so we'll come to them later. So, there is this big population of CD4 positive T cells, and I really want to stress how big this population is. You have a huge number of these cells. Think of how many lymph nodes you have in your body. All of those are full of these white blood cells, these T cells and B cells. And a lot of those will be CD4 positive T cells. So you have a huge number of CD4 positive T cells in your body. They're in lymph nodes, they're in the blood, they're in the spleen. Now, all the different CD4 positive T cells are directed against different peptide fragments. They all have a different specificity, a different peptide fragment that they are directed against and which they absolutely hate. Now remember, we will have CD4 positive T cells against a huge variety of peptide fragments, but we will not have CD4 positive T cells, ideally at least, we shouldn't have CD4 positive T cells that are directed against peptide fragments that could come from a self protein. So we do not want it to be the case that if you take a self protein, and I'll just draw something here, so let's say this is a self protein, we do not want it to be the case that if we chop up this and get a little peptide fragment here, that we have a CD4 positive T cell that's directed against that. We only want CD4 positive T cells that are directed against peptide fragments that you can find in pathogen proteins. And remember, we're dealing with quite long peptide fragments, a lot of amino acids long, 10 to 25 amino acids long, and there are overall around 20 amino acids, so there are a huge number of different peptide fragments that fit those specifications, and in self proteins you will find different sequences of amino acids to those that you would find in pathogen proteins. So it is possible to find peptide fragment sequences that would not appear in any self protein in the body, but appear in the pathogen proteins, and these are the CD4 positive T cells that are going to be important to us. So remember, to avoid autoimmune responses, we must not have CD4 positive T cells directed against peptide fragments that can be found in self-proteins. We only want CD4 positive T cells directed against peptide fragments that are found in pathogen proteins, but not self-proteins. So, we have chopped up influenza viruses' antigens into loads of peptide fragments, and an army of antigen-presenting cells are now presenting these peptide fragments to T cells, trying to find CD4 positive T cells directed against any of these different peptide fragments. Now, what's going to happen is a bunch of these peptide fragments are going to be successful, i.e. the antigen-presenting cells will find a T cell that is directed against one of the peptide fragments that it is presenting. So for a whole bunch of peptide fragments of the antigens of influenza virus, we are going to activate CD4 positive T cells that are directed against that peptide fragment. Of course, it won't work for all the peptide fragments. In fact, most of the peptide fragments will not ever actually work. We won't ever the antigen-presenting cells won't find CD4 positive T cells directed against that peptide fragment, and therefore you won't initiate a successful T cell response against that peptide fragment. But for some of the peptide fragments of some of the antigens of influenza, you will find CD4 positive T cells of that specification, and those will now undergo division process. So they will be activated by the antigen presenting cell, and what they then do is they divide and divide and divide to produce a whole population. And I've drawn 6 here, but in reality it will be far more than 6. A whole population of identical CD4 positive T cells that are all directed against that peptide fragment. So this will be happening for multiple different peptide fragments. For each of the different peptide fragments that are going to be successful, they'll have a different CD4 positive T cell with the specificity against them, and all of them will now be dividing to produce a whole population. So what happens next then? Well, these CD4 positive T cells that we've now got here, an expanded population of them, are going to start differentiating. Now, this is a bit of a complicated story. So, what's going to now happen? They're going to differentiate. So, they're going to turn into effector CD4 positive T cells, or some of them are going to turn into memory CD4 positive T cells. So, most of them are going to turn into the effector cells that are actually going to go and help fight the pathogen, and these are called T helper cells. So let me get another colour apart from green to do this in. Right, so there are loads of different options for what each one of these cells in this population can differentiate into, and different ones will differentiate into different types. Now, let's firstly go over the different types of effector CD4 positive T cells that they can become. So they can differentiate into effector CD4 positive T cells. And there are 4 major different types of effector CD4 positive T cells, known also as T helper cells. So there's another name for the effector CD4 positive T cells. They're also called T helper cells, or TH cells, or just helper T cells. So a lot of these cells are now going to differentiate into helper T cells. So there are 4 main different types of helper T cells that they can differentiate into, and these are called T helper 1, T helper 2, T helper 17, and T follicular helper, which is TFH. Okay, so I hope you're following what I'm saying. Each one of these cells here is now going to undergo a differentiation process, and in this differentiation process, it can either become an effector CD4 positive T cell, most of them are going to become effector CD4 positive T cells, or the other type of CD4 positive T cell it can become is a memory CD4 positive T cells, and only some of them will become memory CD4 positive T cells. So I'll just cover memory CD4 positive T cells because they're a little bit more simple, because there's only one type of those rather than 4 different types. So, memory CD4 positive T cells, it's a very simple concept. If we get infected by the pathogen again, we might want to initiate this same adaptive immune response. If it was the case that all of these progeny of this original CD4 positive T cell that was directed against a certain peptide fragment of one of the antigens of this pathogen were all to become effector CD4 positive T cells, well, these are not very long-lived. Effector CD4 positive T cells do not live that long. They fight the infection, and once the infection is gone, they then die, and they vanish from the body. They're removed from the bloodstream. So they die. That would mean that we'd have wiped out this clone of CD4 positive T cells. We no longer have any CD4 positive T cells left that are directed against that peptide fragment potentially, which is useless. We don't want to have wiped out that clone because it was actually a useful clone. It was directed against a peptide fragment of an antigen of a pathogen that was actually infecting us. So we want to replace this original CD4 positive T cell that we activated to divide and divide and divide so that we could produce effector CD4 positive T cells with its peptide fragment specificity. And that is what the memory T cells do. So some of these will become memory T cells. I'll draw these in this sort of blue colour here, or blue-grey colour here. And these replace these original CD4 positive T cells that we had here. So if you get exposed to the pathogen again, let's say this same influenza virus comes a few years later, what will happen is the whole thing will happen again. The antigen-presenting cells will chop up its antigens, present them on NHC class II molecules, will go to the lymph nodes, and then they can find these memory CD4 positive T cells here that are directed against one of the peptide fragments of the influenza viral antigens. And now, because there's loads of these, it will happen much quicker, and this is the basis of why a secondary immune response is quicker than the primary immune response, because you have more of the CD4 positive T cells that are directed against the useful peptide fragments. So you'll end up, you start maybe with just one cell directed against that peptide fragment. You'll get loads of memory CD4 positive T cells produced, and they perform the role of this. So these antigen-presenting cells coming along for the second time now will present the peptide fragments of these ones and will activate those ones, and then they'll do the exact same thing as the original CD4 positive T cell, i.e. they'll divide and divide and divide to expand the population of CD4 positive T cells that you have directed against that peptide fragment. And again, in this population, some of them will become memory CD4 positive T cells to replace the one that was used, and some of them will go into the effector CD4 positive T cells, which will actually fight the pathogen. So I hope you understand what memory CD4 positive T cells do. They replace the original one, and they can be reactivated just like the original one. Okay, let's now talk about effector CD4 positive T cells. So these are the ones that are going to actually help. Now we have these 4 different types of effector CD4 positive T cell. Now the reason that these are called helper T cells, is the other name for effector CD4 positive T cell, is that they help B cells to activate. They are really important in the B cell response. B cells cannot activate without helper T cells. Now, every single one of these type of helper T cells can help B cells activate. So they can all help initiate the B cell response. And by the way, I just want to write out what this one stands for. Remember, the FH stands for follicular helper, just to make sure that you understand what that means. This is the follicular helper type of helper T cell. Okay, right, so what then is the difference between them if they can all help in the B cell response? Well, they do have slightly different characteristics, and they form the different arms of the adaptive immune response. So not only will they all help in B cell response, but you would choose to have different ones of these depending on the type of pathogen involved. And I just want to go into this in a little bit more detail. So let's talk about this. So remember, at the site of inflammation where the pathogen is present, the cells of the innate immune system, they can tell things about the pathogen that they are fighting. And it is believed that they probably release certain mediators that we do not understand properly as of yet, but they probably release certain mediators into the extracellular fluid depending on what the type of pathogen that they're dealing with is. And that these mediators probably go to the lymph nodes and tell these differentiating CD4 positive T cells that are becoming helper T cells what type of helper T cell to become. So we believe that what type of helper T cells you mainly get from these differentiating cells here is determined by mediators that are draining in the lymphatic vessels to lymph nodes that have been released by the cells of the innate immune system at the site of inflammation. And they are telling the adaptive immune system what type of immune response we need. So let's just talk about these different types of helper T cell. So I'm firstly actually going to talk about the follicular helper T cells because this is the major type we're going to get in influenza viral infection. These are the best, the most diligent at helping to activate B cells. So if you just want a B cell response, which is what we want in influenza, we want to produce antibodies against the neuraminidase and hemagglutinin proteins to inactivate the hemagglutinin protein when the antibodies bind so that the virus can't get into cells. So we want an antibody response, we really want a B cell response. If you just really want a B cell response and don't want anything more fancy on the side, you go for follicular helper T cells. So you let most of these T cells here, these CD4 positive T cells, against this specific peptide fragment, you let most of them differentiate into follicular helper T cells, if that is the case. So if you just want a B cell response, you let them become follicular helper T cells. And in the case of influenza viral immune response, most of them are going to become follicular helper T cells. And of course, some of them will become memory CD4 positive T cells. But we're talking now of the subset that are becoming the effector helper T cells. Most of that subset is going to become the follicular helper T cells. So when do you choose instead to have these fancier ones, T helper 1, T helper 2, T helper 17? Well, you choose those ones if you want something more fancy than just a B cell response. Yes, they will help cause a B cell response, excellent, but they will do other things as well. And against certain pathogens, you want these other things that these ones can do. So these are really involved in more fancy arms of the immune response than just the B cell response. So let me talk about the T helper 1 cells first, then I'll talk about T helper 2, and then I'll talk about T helper 17. As I say, it's not relevant to the adaptive immune response against influenza, but just for a complete picture of the adaptive immune response, I'm going to talk about the other 3 types briefly. Okay, so we'll start off with T helper 1 and then we'll go up to T helper 2 and then T helper 17. So when would you want your effector T cells to be of the T helper 1 type? Well, this is the type of T cell that you want if macrophages are struggling. If they're struggling to phagocytose the pathogen, and the classic example of this is TB. And so I'll just put this here. So T helper 1, I'll put as the title here. So let's just draw a macrophage. So here's a great big macrophage. And remember, the macrophages are the better type of phagocyte of the innate immune response compared to the neutrophils. The neutrophils are weaker than macrophages. So this is a macrophage. This is a powerful phagocyte. Now, in certain types of pathogen, the most famous example being Mycobacterium tuberculosis, the causative agent of TB. So TB is caused by the bacterium Mycobacterium tuberculosis, a very strange type of bacterium. In reality, Mycobacterium tuberculosis isn't actually as dangerous as lots of different types of bacterial cells. It divides extremely slowly for bacterial cells. It's something like it takes 24 hours for a Mycobacterium tuberculosis bacterial cell to divide in 2. When you compare that to E. coli, which can divide every 20 minutes, it seems pathetic. So it doesn't seem like it's that much of a dangerous bacterium. What makes TB such a serious illness and why this bacterium is such a problem for the human race is that it is incredibly difficult for our immune system to kill it. Even though it divides so slowly compared to other bacterial species, which makes it seem as though it's not that dangerous because, you know, its population is going to expand at such a slow rate compared to other bacterial species. It's really difficult for our immune system to kill it. Specifically, macrophages, which are a major mechanism by which we kill lots of different types of bacterial species, struggle to actually destroy it. So, here is the Mycobacterium tuberculosis. It's a rod-shaped bacterium that I've drawn there in red. And it's inside a phagosome here in the purpley grey colour. Okay? Now, so it phagocytoses it into a phagosome perfectly, but then when it comes to actually killing the bacterium, the macrophage really struggles to kill the bacterium, and in fact the bacterium gets the better of the macrophage. It wins. It stops the macrophage from killing it, and you know, this cheeky little thing even starts dividing inside the phagosome, so that you get loads of Mycobacterium tuberculosis cells inside the phagosome, inside the macrophage. So, macrophage is struggling. This is where we call for a T helper 1 response. So I'll just put this here. So we have a macrophage here that is struggling. It's struggling to kill the pathogen. Now, macrophages can use more powerful methods to try and kill the pathogen. So this cell is a killing machine. It has loads of mechanisms that it can use to try and kill this cell. And initially, as part of the acute inflammatory response, it only uses a few of its mechanisms. It doesn't use the more deadly mechanisms. And the reason it doesn't use the stronger mechanisms is because a lot of the killing chemicals that it tries to unleash on the pathogen spill out of the cell and go into the surrounding tissue when it uses these stronger mechanisms, so they end up causing a lot of tissue damage. So overall, the reason macrophages don't use their stronger mechanisms is because it leads to a lot of damage of the surrounding tissue. So, when they first come in, they're very tentative with the mechanisms they deploy, and in order to be able to use their stronger methods, they need to be given permission by the adaptive immune response. And the reason that that exists is as a checkpoint. It's to make sure that this macrophage is not struggling to kill the cell because this cell might be a human cell. So we don't want macrophages going around phagocytosing healthy human cells. We want to make sure that if they're using the horrendously strong mechanisms, that they really are fighting a pathogen here. And how can we make sure that they're fighting a pathogen? Well, if there is an adaptive immune response initiated against this pathogen, then that shows us it's a pathogen, because you don't initiate adaptive immune responses against self-proteins. Okay, and this is what T helper 1 cells have the job of doing. As well as helping B cells, they also go along and give permission to struggling macrophages to use their stronger mechanisms. So let's say that we have a T helper 1 cell here. So here it is, and it's this classical shape with the massive nucleus here, nice round cell with a big nucleus. So this is a T helper 1 effector CD4 positive T cell, and let's say that this is directed against a fragment of this pathogen that the macrophage is struggling to divide, and struggling to destroy. Okay, so some antigen here has a peptide fragment, and this T helper 1 cell is directed against that peptide fragment because we've initiated this extracellular adaptive immune response. And indeed, Mycobacterium tuberculosis is an extracellular bacterium until it gets phagocytosed and then starts dividing in the phagosome. So, even though the cell is really struggling to kill the Mycobacterium tuberculosis cells inside the phagosome, it will still succeed in breaking down some of its antigens, and it will break them into peptide fragments and then display them on MHC class 2 on its surface, because, you know, it's an antigen-presenting cell. So you'll have this macrophage that's struggling here, covered in MHC class 2 peptide fragment complexes, and one of those may contain the peptide fragment that this T helper 1 cell is directed against. So the T helper 1 cell comes along, finds this cell, and if it does have displayed on its surface MHC class 2 with the peptide fragment that it's against, it says, brilliant, you are struggling with the pathogen that I am directed against. I am an adaptive immune system cell. I am directed specifically against a peptide fragment of an antigen of this pathogen, and you are struggling to kill one of these things. So I will bind to you and then activate you. So what will happen is the T helper 1 cell will activate the struggling macrophage by releasing a very powerful molecule that I mentioned earlier on, actually, interferon gamma. So we talked long ago about interferon alpha and interferon beta. Now here's the more famous interferon gamma, and it's released by T helper 1 cells, and it's the molecule that gives the macrophages the signal. It gives them the permission to use their strong, hideous mechanisms to kill the pathogen. I always think of a chapter of Harry Potter when thinking about this. There's a chapter in Harry Potter 4, Harry Potter and the Goblet of Fire, where Sirius is in a cave and Harry has gone to visit him, and Sirius is telling Harry all about the last time Voldemort was powerful, and he's talking about Barty Crouch Sr., and he says that Barty Crouch Sr. fought the dark arts with fire. He fought fire with fire. He gave the auras, which are the wizards they're the police effectively, who go and capture dark wizards. And he gave them new powers. He gave them the power not just to capture dark wizards, but to kill dark wizards. And I always think that this is kind of similar to that. The T helper 1 cell is analogous to Barty Crouch, the macrophage is analogous to the aura, and the T helper 1 cell, Barty Crouch, is giving the macrophage, the aura, permission to use absolutely brutal mechanisms to try and kill this cell. To try and kill the bacterium, the pathogen. Okay, so when macrophages are struggling to destroy a pathogen, that is when you get your effector helper T cells to differentiate into T helper 1 cells, because they can not only help with the B cell response, but they'll also go into the bloodstream, go to the site where the inflammation is present, and they can go and help the struggling macrophages, give them permission to use brutal methods using the interferon gamma. And again, I'll stress that we don't completely understand what it is that determines what type of T helper cells these cells are going to differentiate into, but we think what must happen is at the sites of inflammation, the cells of the innate immune system, such as the macrophages, must release molecules, signaling molecules that go to the lymph node and say what type of helper T cell we need. And if the macrophages are struggling to destroy these, it's easy to imagine they'll release something that will go here and say, we need T helper 1 cells to help the macrophages out. We don't just need a B cell response here, we need this fancier version here where we have these T helper 1 cells which can multitask. Some of them can stay to help B cells in the lymph nodes. Others can come to the site of inflammation and help the macrophages. So T helper 1 cells is a fancier arm of this B cell response part of the adaptive immune system than just the T follicular helper cells. So we call that the T helper 1 response arm of the adaptive immune response. And it, as I say, is a key one against Mycobacterium tuberculosis. That's the archetypal example. So that's the one that I'll discuss in most detail, but I will now just mention T helper 2 and T helper 17 as well. So T helper 2 now. When do we use T helper 2s? Well, again, T helper 2s, they can help B cells, so that's always a function of helper T cells. They can help activate B cells, but they have another function as well. They are used when we have a multicellular parasite. So let me put this here. So I'll just draw a worm, for instance, because that's the typical example of a multicellular parasite. So if we've got some multicellular parasite here in red in a tissue. Now, multicellular parasites are a big problem because macrophages and neutrophils and natural killer cells are not going to be effective against multicellular parasites whatsoever. We know natural killer cells, they're useful for killing dodgy human cells, so they're no use at all. Macrophages and neutrophils, the phagocytes, are they going to be effective against a great big multicellular parasite? The answer is no, they're not going to be terrific at all against a multicellular parasite. They're good at phagocytosing bacterial cells, fungal cells, and virus particles. They are not good against massive great multicellular parasites. For multicellular parasites, we need to recruit another type of cell of the immune system, a more dangerous type of cell that you don't normally just bring in as part of the standard innate inflammatory response. And that's a type of cell called an eosinophil, so I'll draw the eosinophil here. So there is a very dangerous type of cell that is capable of attacking multicellular parasites called an eosinophil. They are called that because they stain very well with the dye eosin. So they like eosin, and eosin is a red dye, so they stain up bright red when you stain them with eosin. They also have a multi-lobed nucleus like the neutrophils, but they don't quite have as many lobes as the neutrophils. They just have a bilobed nucleus. So here is the bilobed nucleus here in red. And because their cytoplasm stains up red and their nucleus doesn't usually stain up red, the nucleus stains up black. but I couldn't obviously draw black because my background is black. So usually imagine this picture has a black nucleus and then the cytoplasm is all red. You can imagine what it ends up looking like. It ends up looking like a sunburnt face with sunglasses on. So that's what eosinophils are often described as looking like, a sunburnt face with sunglasses on. Anyway, so these are a cell of the immune system. It's circulating in the bloodstream. It's a very, very powerful cell. It has horrible, horrible proteins that it can release. Eosinophil cationic peptide and major basic protein. So I'll just write these down because these are its major killing mechanisms. So I think we'll do orange. I don't think I've actually used orange at all anywhere in this video. So here's some orange. So eosinophils, they have granules in their cytoplasm. So I'll put them here. And these granules are full of absolutely foul proteins, one of which is eosinophil cationic peptide. So eosinophil cationic peptide. And then another one is major basic protein. And these are just cytotoxic proteins. They're capable of killing cells. Major basic protein. So these cells are really dangerous. If they were to unleash these proteins on normal human cells, they would destroy the normal human cells. So we use these very, very carefully in the immune system. And we bring them right next to multicellular parasites and get them to release their horrors from their granules onto the surface of the multicellular parasite to try and kill the multicellular parasite. So as I say, you do not just bring these in as part of the normal inflammatory response. In order to get them to come in, you have to first take T helper 2 cells to the site of inflammation, and then T helper 2 cells can release a bunch of molecules. I think it's interleukin 5 and interleukin 13 to call for eosinophils. So, if you've got a multicellular parasite, what can happen is the antigen-presenting cells will grab a few cells off the surface of the multicellular parasite, phagocytose those, present peptide fragments of antigens of those of this pathogen on their surface on MHC class 2, because of course it's an extracellular parasite. They will activate therefore some CD4 positive T cells directed against peptide fragments of that pathogen. And if we're dealing with a multicellular pathogen, what will happen is the cells of the innate immune system will release certain molecules that will go to the lymph node and cause the CD4 positive T cells to become T helper 2 cells. They'll contribute to B cell activation, but they'll also, some of them will come to the site where the multicellular pathogen is, and then when they meet the macrophages that have been trying to kill this pathogen with the peptide fragment that they are directed against on their surface bound to MHC class 2, they'll bind to that, they'll say, brilliant, we found the site where the multicellular pathogen is, and at that point they'll start releasing interleukin 5 and interleukin 13, so IL-5 and IL-13, and these are the molecules that call for eosinophils. So where you have these molecules produced, eosinophils start coming out of the bloodstream and come to that area, and then they'll start releasing their horrors on this multicellular parasite. So just to summarize that again, when you are dealing with a multicellular parasite, the macrophages will do their best to phagocytose some of the cells of that multicellular parasite as well as the dendritic cells, and therefore they can end up presenting peptide fragments of the antigens of the multicellular parasite on their surface. Therefore they can activate the CD4 positive T cell response. They'll release molecules that we don't have a brilliant understanding of to tell the CD4 positive T cell response that we're dealing with a multicellular parasite here, and therefore it would be ideal if you didn't just produce us T follicular helper cells, instead if you produced us some T helper 2 cells that can multitask. Some of them can stay to help with the B cell response, but others can then go to the site of infection. And when they find cells, macrophages mainly, with MHC and their peptide fragment that they're directed against in this tissue, they will know that the pathogen is therefore present, and they'll release their molecules that call for eosinophils. Eosinophils will come in and start attacking the pathogen. So T helper 2 cells overall are the cells that bring in eosinophils to the site of infection. So, that is the T helper 2 response of the adaptive immune response, another arm of the adaptive immune response. Not an arm that's relevant to us, but as I say, I'm just filling you in with a bit of background here. So, finally, to finish off, let's just talk about T helper 17, another one that's not relevant to us. This is the one that we use with fungal cells. Now, fungal cells, the best way of dealing with a fungal infection is with neutrophils. Neutrophils are the beast of a cell for dealing with fungal infections. Now, of course, we're bringing in neutrophils anyway as part of the innate immune response, so I'll just draw a fungal cell, and I'll find some sort of fungal-like colour. We'll have green. So let's say here is a fungal cell, and I think fungal cells do have nuclei. I think they are eukaryotic cells. So here is our fungal cell in green here, and of course it'll be dividing and dividing and dividing. Now, we bring in neutrophils as part of the innate immune response, but we don't bring in enough. We want to bring in absolutely loads of them. We want the tissue to be utterly flooded with neutrophils, because neutrophils are fantastic for some reason at killing fungal cells. And I'll just label this up as a fungal cell here. Okay, now it is the T helper 17 cells that are fantastic at calling for a huge number of neutrophils to a site of inflammation. So, what will happen then is, if we want the T helper 17 response, you'll get, again, the initiation of the CD4 positive T cell response. So you'll get the antigen-presenting cells, phagocytosing fungal cells, breaking down the antigens into peptide fragments, presenting those peptide fragments on MHC class II molecules, going to the lymph nodes to find CD4 positive T cells which are directed against those peptide fragments. Those CD4 positive T cells will then clonally expand and will begin this differentiation process, and if we want them to become T helper 17 cells, what will have to have happened is the cells of the innate immune system at the site of inflammation will have had to release some molecule to tell these cells to now become T helper 17 cells. They will become T helper 17 cells, and these will multitask. Some of them will go to the site of inflammation, whereas some of them will stay to help B cell responses, which we need against these fungal cells because they're extracellular pathogens. Now, the T helper 17 cells will come into the affected area, and again they'll have to confirm that they've actually come to the right area, so they'll have to find macrophages with MHC class II on their surface and the peptide fragment that they themselves are directed against. When they've found that, they've confirmed that they're at the site where the pathogen that they are directed against is, and they will start releasing what molecule? Why were T helper 17 cells called T helper 17 cells? Because they release the molecule interleukin 17, IL-17, and this, when you get interleukin 17 going up in a certain area of tissue, it causes absolutely loads of neutrophils to accumulate in that area. So absolutely loads of neutrophils are going to arrive there. And by the way, I don't think I've ever told you this, but there's an abbreviation for neutrophils, which is NPhi. Just like macrophages, MPhi, neutrophils we can abbreviate to NPhi. So you get absolutely loads of neutrophils in that area. Okay, so those are the different types of effector helper T cells. The T follicular helper are then the boring ones because they just cause the B cell response, but they cause it the best of any of these ones. They're the most diligent. They just stay in the lymph nodes to help the activation of B cells. So these are 3 other arms of the adaptive immune response, ones that are not relevant to us. We are just interested in the B cell response and cytotoxic T lymphocyte response. We are not interested in the T helper 1, the T helper 2, or the T helper 17 response for obvious reasons, because we do not have any of these types of pathogen that I've just been talking about here. Okay, right. So the next thing we want to talk about then is we're going to abandon the extracellular response for now. We have our effector helper T cells, which are directed against peptide fragments of our pathogen. What is going to happen in the case of influenza is you're going to mainly get follicular helper T cells being produced, as well as, of course, memory CD4 positive T cells being produced. For now, I'm going to abandon this response because the next thing that we need to discuss is the fact that these are going to help B cells activate. The fact that the follicular helper T cells are going to help with B cell activation. We will come back to that. I want to finish T cells first, and there's a whole other class of T cells that we have not discussed yet, which is the cytotoxic T cells, the CD8 positive T cells. Therefore, I'm going to abandon this, we'll come back to it, and for now, let's go to the other type of T cell response. So back up here then, and we're now going to do the other arm. So coming off here. So we're going to talk about the intracellular response now. So I'll remind you of where we have got up to with the intracellular response just to set the scene. So we started with the antigen-presenting cells phagocytosing dying virally infected cells, and these were full of the influenza viral antigens. Okay, so what will have happened is they will have broken down those influenza viral antigens into peptide fragments, and they know then that this is an intracellular pathogen because they found them inside a dying epithelial cell. So they'll put those peptide fragments on the MHC class molecule that is going to activate intracellular responses, that is going to activate the cytotoxic T lymphocyte response, and this is MHC class 1. So they become coated in MHC class 1 peptide fragment complexes, where the peptide fragments are from antigens of influenza virus. Of course, you'll also get MHC class 1 peptide fragment complexes where the peptides are self-peptides. They've come from self-proteins. That will happen as well. The antigen-presenting cell doesn't know the difference between a pathogen necessarily and its self-proteins with regards to what it puts on MHC class 2. It does know it better with regards to what it puts on MHC class with regards to what it puts on MHC class 1, it does know it better with regards to what it puts on MHC class 2 because it had to then phagocytose something from the extracellular fluid. Whereas, when it's phagocytosing an intracellular, a whole cell like this, it's going to chop up a lot of those self-proteins as well and put them on MHC class 1. So what I've just sort of waffled through there is the fact that in the case of MHC class 1, you will get a lot of MHC class 1 peptide fragment complexes where the peptide fragment is actually a self-protein, whereas you don't get that as badly for MHC class 2 peptide fragment complexes because you're not phagocytosing the entire cell when you're doing the MHC class 2 route. Okay, that is not a problem, however, that you get these peptide fragment MHC class 1 complexes where the peptide fragment is a self-peptide fragment, because we should not have any T cells that are directed against self-peptide fragments. So it's only the viral antigen peptide fragments that are actually going to lead to the activation of any T cells. So what's now going to happen is these antigen-presenting cells coated in MHC class 1 peptide fragment complexes where the peptide fragments are from antigens of influenza viruses are going to go to the lymph nodes, and they're now going to activate certain types of T cells, the other type of T cells. So let's put this here and we'll go back to blue for this. Okay, so the other big population of T cells is called the CD8 positive T cells. So these are the two big populations of T cells that we have in our body, the CD4 positive T cells and the CD8 positive T cells. The CD4 positive T cells, they are part of the extracellular response. They are activated by peptide fragments on MHC class 2 molecules. The CD8 positive T cells, they are part of the intracellular response and they are activated by peptide fragments on the surface of MHC class 1 molecules. Now, just like the population of CD4 positive T cells, if we were to look at every single CD8 positive T cell in your body, and again you would have billions of the things, they all have a T cell receptor of their own design, and that T cell receptor will be targeted against a specific peptide fragment MHC class 1 complex, i.e., every single CD8 positive T cell is directed against a certain peptide fragment. And that peptide fragment will not be a peptide fragment that appears in any self-proteins, or at least in principle it should not be a peptide fragment that appears in any self-proteins. It will be a peptide fragment that appears exclusively in pathogen proteins. Okay, So what's now going to happen then? We have got loads of antigen-presenting cells coming with loads of different MHC class 1 peptide fragment complexes on their surface, i.e. they're presenting loads of different peptide fragments to the CD8 positive T cells. So what's going to happen is we're going to get loads of matches, basically. Because we're bringing in so many of these antigen-presenting cells and we're presenting overall so many different peptide fragments of the antigens of influenza viruses, some of them are actually going to work. We will actually end up with antigen-presenting cells meeting the right CD8 positive T cell. Because you might wonder, when these CD8 positive T cells are so rare against a specific peptide fragment and they can be in so many different lymph nodes around the body, they might not be in the right place at the right time. They might not be in the lymph nodes that are actually draining the respiratory tract epithelium. But because we are presenting so many peptide fragments of so many antigens of the influenza virus to the CD8 positive T cell response, for some of them it will work. It will be the case that the right CD8 positive T cells are in the lymph nodes that drain the respiratory tract, and therefore that the actual peptide fragment is presented to that CD8 positive T cell and that CD8 positive T cell activates. But again, I admit that a whole bunch of the peptide fragments will not successfully get a CD8 positive T cell response initiated, and it will be maybe because the CD8 positive T cells that could have initiated the response are in a far off lymph node and therefore aren't exposed to the fact that the peptide fragment that it's directed against is actually in a pathogen that is infecting us currently. Okay, but the point is, some of them will work, so you will get a whole bunch of concurrent CD8 positive T cells against different peptide fragments of the influenza viral antigens being activated. Okay, so again, drawing a little picture of a CD8 positive T cell, it's a classical T lymphocyte. It's got a spherical structure and then the massive nucleus. So what's going to happen? Exactly what happened for the CD4 positive T cells. So if it gets activated, so its peptide fragment is one of the peptide fragments of an influenza viral antigen and it's been presented to it successfully by an antigen presenting cell, then it gets activated, and of course what does it need to do? It needs to divide like crazy. We want loads of you all of a sudden. We didn't previously, but we now do. So it divides and divides and divides to produce a whole population of CD8 positive T cells that are all identical to it, which all have the identical design of T cell receptor to it, and are therefore all directed against the exact same peptide fragment as it. So these are still all CD8 positive T cells. And remember, this will be happening for loads of different CD8 positive T cells against different peptide fragments of influenza viral antigens. So we get multiple concurrent CD8 positive T cell responses. So we get division, and then we're going to get differentiation, just like we had with the CD4 positive T cells. Thankfully, it is simpler in the case of CD8 positive T cells. They can differentiate into two types of cell: the effector cells, which I'll have up here, and the memory cells. So just like with the CD4 positive T cells, we want memory cells. So we're going to get some memory CD8 positive T cells. Some of these CD8 positive T cells that we produced in the clonal expansion here are going to differentiate into memory CD8 positive T cells. Not many compared to how big the entire population is, and of course I've drawn 9 here. In reality you want thousands and thousands of the things, not just 9, but compared to the total number that you'll produce here, very few of them go on to become memory CD8 positive T cells, but that's still a lot of them. We'll still end up with a decent number of these because you've produced so many of these. Okay, and what is the role of these memory CD8 positive T cells? It's to replace the original CD8 positive T cell. Again, I'll go over the motivation for this because it's important to understand this. So many people do not understand the memory T cells. You do not want all of these to become effector CD8 positive T cells because they aren't that long-lived. After they've been involved in trying to eliminate the pathogen, they will die very quickly, and that would annihilate this clone of T cells. we'd lose that clone of CD8 positive T cells that are directed against that peptide fragment if that was the case. We need to replace this original one that was sitting around waiting to be activated, and that's what the memory T cells are going to do. They replace this original one, they will circulate around the bloodstream, they'll go into lymph nodes, they'll go into the spleen, etc., and they will wait and wait, and if you ever get infected by this influenza virus again, then the entire immune response can be initiated again, and we'll use these new ones instead of this one. And that's why the immune response the second time, the so-called secondary immune response, is so much quicker and so much stronger than the first one, because it's going to be much easier to find the right clones of CD8 positive T cells, i.e. CD8 positive T cells that are actually directed against peptide fragments of the antigens of the influenza virus, if we've got much more of them, which we will have now, because we're going to produce loads of these, whereas previously maybe we only had one. So we'll end up with much more of these cells sitting around waiting for this infection to occur that are directed against peptide fragments of the antigens of influenza viruses. So I hope that the memory CD8 positive T cells and the memory CD4 positive T cells make sense to you. They replace the original one that launched the response, and you'll replace one cell with multiple cells, and therefore when you initiate the response the second time, it will occur much quicker because you've now got more cells, so the antigen presenting cell will find that cell quicker because there's multiple of them and it can bump into any of them. Okay, so the other type then, the effector CD4 positive T cell. So let's go for another colour. I'll go for another type of blue, a darker, richer, royal blue. Okay, so the effector CD4 positive CD8 positive T cells then. These have another name. They are called the cytotoxic T lymphocytes. So effector CD8 positive T cells, also called cytotoxic T lymphocytes. Now the picture of them is still exactly the same. They remain looking like this, nice round with a big nucleus. So let's think about what these things are. These are cells that are all directed against a certain peptide fragment of the influenza virus antigens, of an antigen of the influenza virus. And of course you'll have multiple different clones of these because we've launched multiple of these CD8 positive T cell responses concurrently. So you're going to now end up with loads of effector CD8 positive T cells, cytotoxic T lymphocytes, that are directed against peptide fragments of antigens of the influenza virus that is infecting us. What are these now going to do? They're going to go into the bloodstream, they're going to go to the site of inflammation, so the respiratory epithelium, so they arrive at the respiratory epithelium, and then what they're going to do is they're going to kill virally infected cells. They are the better version of NK cells. They are very, very powerful. Now remember I said earlier on, the NK cells, the reason they're quite bad at killing virally infected cells is they're very, very tentative because the signal that they're using is a little bit ambiguous. They are just looking for low levels of MHC class 1, and they need quite low levels of MHC class 1 before they are sure that the cell is virally infected and they have the confidence to kill that cell. If you've just got slightly reduced MHC class 1, it doesn't see that as a big enough sign that this cell is virally infected. And it has to be tentative because you don't want it to go around killing innocent cells. Whereas these cytotoxic T lymphocytes, they go around looking at cells and they get a signal that tells them absolutely straight away this cell is virally infected, and therefore they can go and with complete conviction and just kill the cell. So they are far more likely to kill a virally infected cell than natural killer cells are, because natural killer cells need a really extreme signal before they will actually kill the cell, whereas these need a much less extreme signal in order to kill the cell. So I hope that makes sense. So how are these things going to find cells that are virally infected? Well, it's through the MHC class 1 molecule. So these have come into the inflamed tissue and they're now going to look for virally infected cells. How are they going to find them? Well, let me draw a virally infected cell. So let's say this is a ciliated columnar epithelial cell that is virally infected. Now, earlier on, I told you that all nucleated cells have MHC class 1. So this special molecule that we've seen its function over here, it's not just on antigen-presenting cells, it's on all nucleated cells. MHC class 2, this one over here, is not the same. It is only found on the antigen-presenting cells, so you will not find MHC class 2 on all nucleated cells, but MHC class 1 is not just a special molecule of antigen-presenting cells, it's a molecule used by every single cell in the body that is nucleated. Now, what is the function of this? Well, all cells, all nucleated cells with MHC class 1, they continuously chop up a few samples of their own proteins. So they continuously take samples of proteins from their cytoplasm, chop them up, and then put the peptide fragments on their surface bound to MHC class 1. So I'll just draw this here. So here is an MHC class 1, obviously completely out of proportion here, so I'll just label this up. This is MHC class 1 here. So, what will it have in its peptide binding groove? Well, it'll have a little peptide here. So here is a little peptide, and usually what is happening is all nucleated cells are doing this. They're taking samples of proteins from their cytoplasm, cutting them up in a random way, and putting the peptide fragments on their surface on the many MHC class 1 molecules that they have on their surface. And this is basically telling cytotoxic T lymphocytes that might come into the area about the proteins that are in this cell. Now, if all of these peptide fragments are peptide fragments of self-peptides, then this will cause absolutely no problem for this cell. So if this isn't virally infected, all of these peptide fragments will be self-peptide fragments, and you will never, ever, ever produce, well, you should never, ever, ever, actually it's not the case that you will never, ever, ever, people with autoimmune diseases do, but in principle you should never, ever, ever produce effector CD8 positive T cells, cytotoxic T lymphocytes that are directed against self-peptide fragments. So this cell is completely safe. However, if it becomes virally infected, and I've got into the habit now of showing virally infected with a red cross for it. So if it becomes virally infected, then the cytoplasm will be flooded with viral proteins, viral antigens, and the cell continues to do what it does always. It samples proteins from its cytoplasm, chops them up, and puts the peptide fragments on its surface bound to MHC class 1. And now what's going to happen? The viral proteins are going to be sampled just by random chance. So it's going to end up putting on its surface loads of viral peptide fragments bound to MHC class 1, and this is the way that cytotoxic T lymphocytes can tell that this cell is virally infected. Remember, each cytotoxic T lymphocyte will have a certain peptide fragment of an antigen of the influenza virus that it will be directed against, and if it comes up to this cell and finds that this cell has on its surface that peptide fragment that it absolutely despises and is specifically targeted against, bound to MHC class 1, it will say, my goodness, I found you, and it will say, I'm going to kill you. This is the signal that this cell needs to say that this cell is absolutely virally infected, and you will see that it is a much more convincing signal. This instantly tells this special type of cell that this cell is virally infected, and therefore with complete conviction it can kill this cell. So, let me just draw a little picture of this. So, I want my royal blue back again for a moment. So, here is the cytotoxic T lymphocyte coming up here, like so. Here's its big nucleus. And what, of course, will be binding to the MHC class 1 with the peptide fragment will be the T cell receptor, here in blue. So that is the T cell receptor. And when this happens, this tells the cytotoxic T lymphocyte that this cell is virally infected, so it uses killing mechanisms. Now, what killing mechanisms does it use? Well, one of its killing mechanisms is actually the same as the natural killer cells. It uses perforin and granzymes. So it releases granules that will contain perforins and granzymes. And remember what happens. Perforins are oligomerized to form a pore in the cell membrane, whilst the granzyme enzymes go through that pore into the cytoplasm, initiate a cascade which leads to apoptosis of the cell, so the cell dies. But the cytotoxic T lymphocytes also have another rather funky mechanism, which is that they can activate apoptosis from cell receptors. So Cells have on their surface, again, all cells have on their surface, all nucleated cells have on their surface a receptor, a very scary receptor called the FAS receptor, and God only knows what it stands for, but this is the FAS receptor. Now, what does this receptor do? This is the receptor that, if enough of them get activated on the surface of a cell, will make that cell commit apoptosis. It is the receptor to die. It is the receptor to commit suicide. So clearly this is a very dangerous receptor, and only very powerful cells should ever have the ability to activate this receptor. And cytotoxic T lymphocytes are granted that privilege. So cytotoxic T lymphocytes have on their surface the ligand for this receptor, which conveniently is called Fas ligand. So FAS-L for short. So this is Fas ligand. So this stands for ligand. So on the surface of the cytotoxic T lymphocytes, you have the Fas ligand. This will bind to the Fas receptor and trigger this cell to commit apoptosis. So just like the perforin granzyme, but for a different receptor through these extrinsic receptors. And this is called the extrinsic apoptosis pathway. So I'll just write that down. So the Fas receptors activate the extrinsic apoptosis pathway of the cell. So all nucleated cells have the ability to undergo the extrinsic apoptosis pathway. They all have the Fas receptor on their surface, which can in principle be activated by Fas ligand. Thankfully, very few cells have the ability to use Fas ligand. Cytotoxic T lymphocytes are one of them. So if cytotoxic T lymphocytes bind to the surface of a cell because their T cell receptors can bind to MHC class 1 peptide fragment complexes on the surface, that means that the Fas ligands will be able to then bind to Fas receptors, and this cell will commit apoptosis for that reason as well. So two killing mechanisms here, perforin granzyme and Fas ligand. Fas ligand isn't used by the natural killer cells. This is a mechanism reserved for these cytotoxic T lymphocytes, which can be absolutely sure that the cell that they have come across is virally infected. Okay, so that is the cytotoxic T lymphocyte response utterly completed. What we've been through, we've completed this arm of the adaptive immune response. That's a nice short story there. Whereas the B cell response is a longer story, and we haven't finished it yet. We've seen the activation of the helper T cells. In the next video, we will go on to the B cell response.